Okay, good afternoon everyone. It's a pleasure to introduce today's speaker, Darshan. Ran is an alumnus of the ICNC, completed his PhD here at ELSC, working on theoretical models of learning in neurodynamics with David Hansel. After that, he established a group as a research fellow at Geneva Research Campus, and recently returned to join the faculty at Tel Aviv University. He's now affiliated with the Department of Psychology and Physiology and Pharmacology, the School of Neuroscience, and the School of Physics and Astronomy. Quite an interdisciplinary position. Ran's research focuses on understanding how neural populations give rise to complex behavior using tools from theory and data-driven modeling. In today's talk, we will present a new line of work exploring the causal role of population-wide neural dynamics in decision-making, and specifically what can we learn from the dimensions of neural activities that are not directly aligned with a typical task relevant code. Please join me in welcoming on.
Sounds like a ChatGPT. Right, so it's fun to feel good. Right, so thank you everyone. I want to say that I'm happy to be here again, but David told me don't tell it. So, but really, I'm happy to be here and happy to meet old friends, but also new friends that I met today. And I'll talk today about data-driven models that we apply to decision-making tasks. This is kind of it's unpublished work, and we are closing that now. So I think it's a good opportunity to get some inputs. Typically, I like to show things that are already closed, but I think this is a good environment to get some inputs. So we'll do that. Before that, let me give you some kind of the philosophy of the things that we are doing or want to do in the group. And you know, we all in computational neuroscience and system neuroscience, we have this kind of general goal that we want to understand kind of how structure will give rise to the neural activity that gives rise to the behaviors or the function. And at least with respect to when I started the PhD, we were really in excited times because we start to have a lot of data sets and a lot of understanding on this, maybe not understanding, but a lot of data on the structure, the neural activity, and the behavior. So for example, when I started, we knew more or less the connectivity of the C. elegans. Now we start to have all kind of data sets with all kind of resolution that we start to know some things on the structure of the network. Here is an example from Janelia. This is it's the mouse light project where we know a bit of the connectivity. So each color here is one neuron, and we know the axons where they go, and we can get some mesoscopic connectivity. And the second thing is that, you know, there are lots of methods now to record for many neurons. And many neurons could get to millions in behaving animals, in like the zebrafish. And here's an example of NeuroPixel records. This is the data I will talk later. This is from the Svoboda group. It's about 60,000 neurons recorded. Here about thousands of them simultaneously from multiple brain regions. You see all the complexity in your eyes, kind of, these are neurons that are locked to the leakings. There are neurons in the thalamus. They are locked to the response. We'll talk about the behavior later. But it's really an impressive data set that we start to have multi-regional at the level of single cell, at the level of milliseconds, which is typically what we want if you want to study decision making. And finally, also the behaviors, right? We really have a good quantification now of the behavior. Here's an example using deep lab cut where you have rodents that is kind of doing this nice reaching task, and we can measure each one of the joint of the fingers and use that in our modeling, in our way to understand how this W, this connectivity, gives rise to the neural dynamics, gives rise to the behavior at the end. So we are in these exciting times, and if you're doing models, you have this complexity in your face. It's a bit harder to do models, but you still try to do that. And when you do models, you're putting yourself in this imaginary line of abstraction, using more complex models or more abstract models. And of course, we like to do abstract models because we can do some mathematical analysis or something like that. And because they have this imaginary line, meaning telling us that the more simple the model is, the better we can understand it. But at the same time, if we go to more and more abstract models, we start to lose the ability to predict the neural activity, to predict the data. So we have this imaginary line of predictive power that goes down, and we would like to be somewhere in this sweet point in the middle. But with all of this data set of the complexity of the structure, the complexity of the neural activity, and the complexity of the behavior, I would argue that what we really want to do is really to push this boundary. We want to take this to go into a more complex models that we can use to predict something in the behavior to be maybe hopefully quantitative, which is almost like a word that you are not allowed to say sometimes in biology, but we are there. I think we can be quantitative, but still have some level of understanding. And that's kind of what we try to do. And I'll give you some examples. Really, everything is published. I also gave talks on that. Last year I gave talk on one of these examples. So it's just to give this taste, and then I will go to our unpublished work, which I'm really excited about, and this is the thing that I would like to talk about. So here's an example from our work with Sasha Rifkin when I was at Janelia. The idea back then was that if you go and you look at tuning curves of neurons, for example, in the head direction system, so you see these nice tuning curves of the neurons, and the way to model this stuff was typically with continuous attractor models, which are models that are built on some symmetry that you have in the connectivity, that they give rise to these tuning curves, or what we call continuous attractors. But if you go now, when you start to have more and more data, we start to see that these typical neurons are not that typical. And if you go to the supplementary, for example, of this paper, you see that there are many other neurons that have multiple peaks. They have high firing rate, low firing rate. There's a lot of heterogeneities in the data that we tend to ignore when we do this model. But this is an example of a model that completely failed to explain this. It is built on this kind of symmetry principle that you have in the connectivity that give rise to symmetry in the activity, and you don't see here any symmetry. You see here neurons that maybe have different preferred direction, but they also have multiple peaks, and they have high firing or low firing rate. Similarly, if you look at the activity at the manifold, right? What is the manifold? I look at the activity of the neuron, I project it, or I don't need to project it, but here is an example. We project it to 2-dimension, and this is the head direction system, so when animal moves, then the activity is kind of moving on this manifold. And you see that this manifold, it seems that it is kind of this one-dimensional manifold that you expect from this system. It has a topology of a ring. But if you go to the data, you see that it's not clear that it has a geometry of a ring. There are areas, I don't know what is this Spitz area over here, but there are areas that maybe are more represented. There are areas that have some curved, they are a bit curved, and these are something that I don't know how they affect the computation, but we really cannot get them in the classical model. And with Sasha, what we did, we developed a new type of models. They are not built those symmetries. In some cases, you can have kind of a hidden symmetry, but in general, there is no symmetry. And then you can generate tuning curves that are highly heterogeneous. You can understand how the heterogeneities in the tuning curves, how they affect the computation, how they affect your ability to integrate your inputs. And you can generate all kind of weird attractors. Maybe this is kind of this manifold that we see over here. And it turns out that these kind of areas, this Spitz that you see over here, the area which are highly curved, the dynamics around them is very different, and it's less stable, it affects the functions, it affects your ability to integrate. So there's a lot of things on the function that comes from this type of manifolds that are a bit maybe more biologically relevant. Another type of work is about the subthreshold activity. When I came to Janelia, and they had this kind of voltage imaging that they started to have. I remember I met Yoav Adam there, and he was afraid, oh, maybe this is kind of, they're going to scoop us. I don't know, but they had this nice voltage imaging. It's still not that good. I didn't work on, like I worked on this data, we didn't publish anything. But this pushed me to look a bit on heterogeneities in the subthreshold activity. So typically when we do modeling, we are ignoring the subthreshold. We are looking at the spikes, we are looking at the rates, we are looking at the inter-spike interval. But if you went to the subthreshold activity, so here's an example of wholesale recordings from anterolateral motor cortex, we talk about that. And you see that some neurons, there are 4 cells over here, and some neurons that are close to threshold, others are far from threshold. That's typically what you see in the data. If you take the typical model that we all use, kind of strong inhibition, strong excitation, and the neuron is bombarded by many cells, so it's opening many channels, and it operates in this high conductance regime, and all the neurons are clamped. All the neurons, they have very little heterogeneities in their subthreshold activity. And a way to solve that, one way to solve that, what we showed, is kind of to take into account the morphology of the neurons, and again, in the network, in a phenomenological way. And again, if you're interested, we can talk about it later. Finally, this is something that we now use a lot. We are fitting models to data, and this is what I'm going to talk about today. And this is something that I actually talked about last year when I was here at ELSC. It's about a mechanism for distributing task-related activity in the cortex through task-independent synapses. But again, I talked about that, but that's another example. All right, so what I want to talk today is this new type of work that we're doing. This is a work together with Ulysses from the Allen Institute. There's a scientist there. With Karel, he's also in the Allen now. Susu, she was a postdoc at Janelia. I think, she was working on this data set for kind of 5 years, I think. And I really wanted to put my hands on this data set, and it took me a lot of time to do that. We published one paper on that. This is the second one. It's a very rich data set. I'll show you the data set in a few slides. So we are doing this work, and as I said, we're closing that. Musi and Shani are students in the group, and they're not directly related. They're not directly part of this work, but once in a while you might ask me a question, and I'll tell you, this is a good thing. We are a good question, and we are thinking how to solve it. So when I say we, it's me, Musi, and Shani, and also Ulysses. All right. So that's what we're going to see. We're going to see the first part, kind of a new type of data constraint mechanistic models that we developed. And we're going to apply these mechanistic models to data, to decision-making data. We're going to extend this framework to single trials, so we're going to apply it to single trial data sets. And then, if we'll have time, we'll talk about multi-regional interactions. So we'll feed the model to the motor cortex, the anterolateral motor cortex, and the thalamus. All right. So let me kind of zoom out a bit and first describe to you kind of two approaches that we have in the field to understand how this complex neural activity is related to behavior. The first approach is kind of this statistical approach. It's dimensionality reduction. So if you think about neuron over time, then we can think about a point in time as a vector in n dimensions. N is the number of neurons. And when time evolves, then this vector evolves over time. So this would be kind of a trajectory in this neural space, in this n-dimensional space. So this would be the vector arc. Now let's bring in the task already. So the task we are exploring, this is this delayed response task, or memory-guided decision-making task that Svoboda was studying for the last 10 years, I think. It's a super simple task. A mouse is head fixed, and so you can record from its neurons, and it's instructed to link to the left or to the right. Here is by a low or high auditory tone. It needs to keep this information in memory for about 2 seconds, depending on the task. And then he responds by linking either to the left or to the right. So if you think about the trajectory of neural activity when the mouse links to the left, then this is this red trajectory. But when the mouse links to the right, you can think that there is a different activity in some area that we record. So the activity will look like a different trajectory. You can look at one single neuron, the tuning curve of a neuron. This is a neuron in the anterolateral motor cortex, so I will call it the motor cortex. And you see that this is kind of the projection on one neuron, right? Just the tuning curve of one neuron. And a neuron is kind of ramping its activity before the go cue, and it's selective. These neurons, for example, is firing more when the animal licks to the right than when it licks to the left. Now, what we can do, and when I say we, what people in the field are doing for at least, I would say, 10 years, we can calculate some dimensions that are interesting. What do I mean by that? For example, instead of looking at the single neuron, we can look at the population activity, and we can look, for example, at this choice dimension. So think that anytime the animal is looking to the right, there is this neural trajectory. When it clicks to the left, there are other neural trajectories. There are very many trials, and I can ask, all right before the cue, what is this direction, this dimension in this n dimension that is best separate the left and the right? So I can do that. This is called kind of the choice dimension. And I can project the activity on this one-dimensional vector, which is the choice direction. And that's what we see over here. This is called the projection or the latent, and the vector is sometimes called the factor. And you see that activities start to separate, and it's highly separated right before the go cue. And this is kind of a way to do this simple dimensionality reduction, correlating this activity in this neural direction to the behavior, which is the choice. Now we can do the same at a different time point. So maybe right before the go cue, we calculate this choice, this coding dimension that we call choice. We can do that right after the go cue. And this we'll call it the response factor or the response mode. And you see that this mode is indeed best separating the left and the right trials when the animal is making a choice. And there are different ways to do that. Other ways are kind of just regression that many people are doing. I think the most famous paper is
zero for this response.
Yeah.
What is zero then? Is it go cue?
Zero is a go cue. It's time for go cue.
Zero
Is a go
The
cue.
leak.
What is that?
And here this is the zero, this is a leak.
This is the go
This
cue.
is the response. This is the go cue.
This is the go cue. Okay. Now we can. So this is one way to do that. Other way that many people are doing is just regression. We can regress the neural activity. We can regress the neural activity and the behavior, and do the same, right? We will have this factor that we'll call the choice, and we project on that, or the stimulus, we project on that, and now we project it on 2 dimension, not on 1 dimension. And then we can see, so this is the famous behavior that is studied in Bill Newsom's lab of this drifting, this random dots decision-making task. And you see that, when we project it on the choice, for example, then when the animal go to the right, the activity projected on this 2 dimension goes to the right or goes to the left if the animal is doing a left decision. In other words, we are spanning. This dimensionality reduction means that we span the neural activity with the choice vector or the stimulus vector or whatever coding that you are interested to study. And the latents are just these, these coefficients in the beginning, and they change in time when we do this dimensionality reduction. All right, so this is really cool, and we learn a lot about that, but you need to remember, it's not, this dimensionality reduction is not grounded on a mechanism. And we want to have something which is really a mechanism. We want to understand how neurons interact to operate, to generate these dynamics. And the second thing that people sometimes forget is that it's correlation, it's not causation. So it's like tuning curves. Tuning curves are not, they're not the way they're correlated with the behavior, and that's it. Right? They're correlated with the behavior. It doesn't mean that they are causally driving the behavior, and so you just need to remember that when you do that. There is a different approach, and we're going to combine these 2 approaches. So there is a different approach that comes from this computational neuroscience field or theoretical neuroscience, which is to look at this high-dimensional neural activity and try to build mechanistic models of neurons that interact and go from this high dimension through this what is called the mean field analysis, kind of to find what are the sufficient statistics, or we call it the order parameters. What are the variables that are interesting to study? And the example that we can give is this one model where you start with this kind of 10,000 neurons of 2 populations that compete, a non-selective population, inhibitory population that interact together, but you can do some analysis and you can derive from that the dynamics on only 2 populations, and you can connect the parameters from these 2 populations to the parameters that we have in the general, in the high-dimensional system. So this approach kind of tells us, all right, let's go from this high dimension to a low number of latent variables, like we do in the dimensionality reduction, and our hope is that this P is sufficiently small. And if P is sufficiently small, maybe you can analyze that. This is a classical example of these 2 populations. You see that we have kind of this is kind of the latent. This is population 1 and 2. We have one attractor, another attractor, and the activity starts, and if it's a left, you go to here, or if it's right, it goes to here, and it's left, the trajectory goes to here. So you have this nice framework of how decision makings are made from a mechanistic point of view. And under some assumptions, you can even reduce this 2 dimension actually to this axis, to this 1 dimension. And then you kind of have these 2 attractors with this barrier between them. And this is exactly our choice dimension that we described before. This is the dimension that if I'll best separate the left and the right halves, right? So this dimension will be these dimensions here, and in the 1 dimension, it's this kind of 1-dimensional axis that describes the difference between the attractor. Now this is also really cool, but the problem is that it's not data-driven. We cannot capture really the statistics that we see in the data. And there is really minimal heterogeneity. So, I'm not showing you, but if I show you how this activity looks in the data, there's no dichotomy. There's never 2 populations. It just doesn't exist in the data. You have a distribution of left and right populations in some sense. Most of the neurons, they are not selected, and there is a continuum of them, and if you put your p-value somewhere, maybe you find 2 populations, but there are no 2 populations. And finally, everything is stationary. So if you have some time-varying activity like we have when we have decision making. So when I make a decision but I'm tired because I talked a lot before this talk, then the activity in the brain will probably be very different than if I'm engaged. And this is not captured here. You can maybe add it, but it's not captured. All right, so that's what we are going to do. We are going to combine these two methods and develop a model to do that. And we're going to study the role of the residual dimensions. So what are the residual dimensions? I remind you we can spend the activity of the neural activity with these coding dimensions, but it's only approximation. There are actually other dimensions that we are throwing away. Right? This is kind of the dimensionality reduction. There's actually some, some orthogonal subspace there that we need to remember. In this model, in this dimensionality reduction, it's kind of this, whatever is orthogonal to this board, to the screen. And here, maybe in the models, it's kind of this non-selective population that we averaged out when we did this mean field analysis. So our question is going to be, can the residual dimensions, so they're orthogonal to the coding dimensions, and you see that actually they explain a small amount of the variance in the data, can they actually shape the neural dynamics and the computations? And of course, if I'm raising this question, probably the answer is yes. So that's the take-home message of what we are doing. And we are studying these residual dimensions using all kinds of these methods that we of fitting that models to data. All right, so how does the model work? The model is super simple. It's a rate model. Like the vector R decays in time, we have some nonlinearity function, and we have a W matrix, an input from outside, and we want to fit this model to the data. We have neural activity over time, and we want to fit this model to the data. And what we're going to do, we're going to take this recurrent connectivity W, and we're going to decompose it to a low-rank, it's going to be a low-rank matrix. What does it mean? It means that we can decompose it to p vectors, so the rank is going to be p. And again, if you don't know, these cartoons are, people like to draw that to explain that this is a low-rank matrix. If you know that, it's clear. If you don't know that, then maybe it's better to have equations. But stay with me, and I'll grab you back in 2 slides if you are not following. But it's super simple. We have p vectors that we are going to train. And in order to replicate the activity of the data, some of these p neurons are going to be the coding dimensions. I'll tell you in a slide or two how we choose them. The rest of them will be the residual dimensions. I'm going to use them. I will need them to explain the data. And there is this small connectivity matrix that tells me how these dimensions interact with each other. So this is this matrix A. So in a way, it's kind of this dimensionality reduction or this in-field approach that we go from this n-dimensional neural activity to p order parameter or p variables, p latent variables. And this A matrix tells us how these p variables interact with each other. And you might think that this is kind of, why did we do that? I want to first kind of make a joke that this is explain kind of the 90% of the models that we have in the field. Here's an example. If you just takes this vectors U to be random vectors, this will be the memories in the Hopfield network. And this A matrix, if you take it to be diagonal or maybe something small on the off diagonal, this is just the Hopfield network. It's kind of a vector that is pushing back the activity and it generates an attractor dynamics. This is how you generate attractors. You can have Chaim had this paper on how you generate sequences. It's kind of, you have kind of this shift matrix over here. It generates sequences. You can generate a ring attractor with these 2 vectors. You take them to be sine and cosine. You can generate it, and so on. And many people, including us, we are training these networks. We are trying to understand, how they work. And so this is really explaining at least a lot of the attractor or the sequence models that we have in the field. Okay, so what are we doing? We take this model and we're going to train it to the data. What do I mean by training to the data? The first thing, we are going to choose the vectors that we want to be the coding dimensions. How do we do that? This is this kind of what we described before, this targeted dimensionality reduction. For example, in our task, it will be kind of the choice direction, which is the direction that best separates the left and right right before the go queue. This is one vector in this n-dimensional space. Another one is what happens. Another one is what happens after during the stimulus. So this is a kind of population that best describe the auditory tone, if it's high or low, and what happens right before the go queue. So these are how we chose the coding directions. And then we're going to have a loss function that tells us, all right, we want to have a reconstruction loss. We want, at least at the first stage, that the activity of the neurons in the model will be like the activity neurons that we see in the data. So this is a trial average loss. We have orthogonalization loss. This is kind of a detail point, but we need that. And we have the coding loss, which is kind of a soft loss that tells us, all right, and can we choose more or less the coding dimensions to be what we want you to, what we extracted from the data.
What about the inputs?
The inputs? Yeah, so there is one input which is also training, that's true. One input is coming through the sample, and in this one-dimensional, this model where you don't have a thalamus, etc., it's just coming here, it's a random vector that you also train, and it kind of in one direction or the other. And a response as well. It was kind of an unselected input, which is the go queue. Yeah.
How did you choose P?
Yeah. So maybe we'll get to that. Okay. And then if the answer won't be okay, we'll get to it. And finally, so we're going just to change the network parameters. How many do we have? N. Right. Each vector is n times p. And we have p squared here, but it's an order of np. And also we learned so that's more or less kind of n times p parameters. So the learning is also quite fast. All right, we're going to apply to the data. And we're going to start with anterolateral motor cortex and only trial average, and then we'll go to single trials and we'll go to multi-regional. So that's the data set I presented at the beginning. It's neuropixel probes. Here you see all the locations and all the numbers of the neurons that were recorded. This is really a massive data set, extremely rich, both a lot of neurons, but error trials and the behavior and everything is even to extract kind of this data, you need to know what's going on here. And so first of all, you have about 66,000 neurons. And we have 28 mice, more than 150 sessions of multiple probes simultaneously. From ALM, all the neurons are about 8,000. We have for left and right hemisphere. We train it now separately. And we have multiple sessions with more than 100 neurons. So when we go to the single trials, this is going to be also important. So we have about a dozen of sessions with 100 neurons. Only within ALM, right? Between the areas we have more. And at the end, we'll also train a network with the thalamus. So we have about 2,000. We have a lot of neurons in the thalamus. We have about 2,000 of them in the ventrolateral, in the ventromedial thalamus, which are we currently connected. With ALM. All right, so here's the model. It was trained to the data. You see here 12 neurons. You see that neurons are selective, right? This neurons, for example, it's the tuning curve of the neuron. And the solid line, the thinner line, is the data. The dashed line, the wide line is the model. And you see the model is doing quite a good job. It's not kind of overfitting this crazy things, or these fast things, but it's doing a good job, I think. And you can look at the projection on the coding dimensions, right? This choice stimulus, the choice dimension, the stimulus dimension, the response dimension, and again, both the data and the model are doing a good job. The data is doing whatever it's doing. The model is doing a good job. And you can also look at the residual dimensions, which are the other dimensions that I didn't tell the model to fit, but the model needs them in order to fit the single neurons. And you see that the model and the data are also, again, they're doing a good job. Now, here's an interesting result. The dimensionality in ALM is super, super, super low. If you look at the PCA, either of the right or left hemisphere, with 2 or 3 dimensions, you already explain about 90% of the data. if you train the network and you have you take these coding dimensions or maybe a little more, again, with 3 dimensions, you can explain 90% of the data. If you want to explain most of the data, or I don't know, if you want to saturate, you need 10 dimensions. And this is the number that we take because of this. But you see that these residual dimensions, they explain about 7% of the data. They're really 7% of the variance. Sorry. Here's how it looks like. If we train with only 3 vectors, then it looks okay. This is the dash, but still you are missing some of the neural activity. Here you are missing a lot, right? You are missing the selectivity of the neuron. But in general, I think these are also bad, like the neurons, they don't look that good. But with 10, you capture everything. So these residual dimensions that we throw away really explain a small part of the data. And here comes the take-home message, and then we're going to show it in different ways. And what happened in the model now if you perturb this choice direction? If it's just a one-dimensional model or this two-dimensional, the 1-1 model, and if you perturb along the choice dimensions, you're going to move above the separatrix and you're going to flip the decision. But you see that if you are perturbing along the choice dimensions and we project along the choice vector, and you see, and again, perturbation is this wider curves. You see that there is some perturbation, but there is a recovery back to the choice or to the activity that was before the perturbation. If you look at what happens in the residual, the projection in the residual, where there was not a lot of difference and nothing really happened. So the choice direction is quite stiff, and it's hard to perturb it. It's hard to flip the decision. That's not the case for the residual dimensions. So for the residual dimensions, one of them, I chose one of them here. There are 7 because I take 10. And you see that if I perturb and the perturbation is here, if I perturb this mode, then I can flip the left trajectory to be a right trajectory. So I'm generating an error in the network by this perturbation. And you can project also on the residual. You see there is some activity that is, there's some deflection over here, but this residual goes back to what it was before. So the take-home message from here is that there is this choice direction, which you might think that that's the way to flip the decision. Well, it seems to be a stiff direction. Other directions that explain about 7% of the variance, they are flipping the decision in a dynamic way. Systematic, yeah.
Your model here is very different than the Montesquieu. There's no evidence that you use. So this whole choice direction is something you invented. There's just 2 points in space.
What do you mean by that?
Now 2 points, 2 attractors, and you decided to put a line in. But there's no attractor or attractive line between them like the Montesquieu.
There's no what?
There's no line attractor, for example, like in the Montesquieu case.
No, I didn't say that
So
there's
why
accumulation of
do you-
evidence.
so
I
why
didn't
would
say
you expect to have some line and a gauge in between on this line? There's nothing happening.
So I don't know if there are.
Some line we invented in space, or
It's a line that I invented. All right, so let's take it to the
it
one.
separates 2 points.
Let's take it to the 1 neuron. All right. In 1 neuron, you will see neurons that is active when you look to the left, and is active very little when you look to the right. You will say, this is a selective neurons. It's tuned to the text, right? In the n dimension is the same, right? There is a direction in space that's best separate right before the go cue, the left and the right. So it's not 1 neuron, but it's a different dimension.
Is not along that line.
Right?
But the dynamics is not along that line.
So when you do this, that's what naively you expect. I agree with you. In this 2 dimension, it is, right? It doesn't have to be the case. But typically, here's another thing. That typically, when you have these two population, and the input, the stimulus direction is doing that. And the choice is doing that. So the stimulus direction and the choice is in the same direction. In the data, they are orthogonal. So it's really not these 2-dimensional properties. And now you're right that I can build somehow a different model. That's not the classical one. And what we see over here is that the dynamics is actually not 2-dimension. It's low dimension, but it's actually it's higher dimension. And at the end, it will end into, and we'll show that, it will end into attractor dynamics. But there is kind of a transient dynamics that ends into attractor. This is very, very different from the classical way that people think on decision-making and these models. And I didn't say there is accumulation of evidence and
classical decision-making are not network models, they are phenomenological.
okay, so you can go to phenomenological model of diffusion. I agree. I'm talking about the mechanistic models that we have. Kind of this one model is kind of a classical one. But I agree with you, the dynamics, there is dynamics here, and that's what we show. Yeah?
So how do you reconcile this result with thinking about the Charles' work with the perturbations where the network seem very robust in the data? You can throw it pretty much anywhere and it returns.
So maybe I'll show results on that paper, I think. I'll show results afterwards from the experiments, and then we can go back to that, okay? Okay, but now we are still in the model. We can now do it in a systematic way. We can go in each one of this direction, and we can perturb it more strongly or more weakly, and in the other side as well. So this is what you see over here. You can perturb it here, strong or early, doesn't matter. Sorry, strong or weak, or in the other side, doesn't matter. You have this recovery. And if you do it in this residual dimensions, then you can flip the decision. And you can do that later, and you can perturb the activity during the delay, but later on. Still, you can kind of flip the decisions. And here, you don't see that in the choice. But later on, right before the choice, there is kind of already kind of commitment for the choice. From the dynamics, it looks like that all directions are becoming stiff. And the way to quantify that is the rate of flipping the choice in the model as function of the perturbation time. If you perturb any of the residual or the choice, and you see that all goes down. So your ability to flip the dynamics goes down. And this gives rise to this idea that maybe there are these attractor dynamics, and that there is kind of a high-dimensional trajectory that at the end goes to this attractor dynamics. And a way to test that, or one way to test that in the model is very simple. You just take off the GoQ. So with the GoQ, it looks like that. Without the GoQ, you see that this is the dashed line. You stay in the right place, and you can calculate the Jacobian and show that this is a stable point. And you can go also to in the model and give a catch trial. This is something that people typically do in monkeys and humans. Now also they started doing that in mice. And the behaviors in mice, I think, is always in the lag into monkeys. You can see that if the catch trials, if the GoQ comes up to 600 milliseconds, then you still stay in the same place. And if you go to the data, you have some catch trial in the data. You see also that it seems that the activity is not collapsing if there is in the catch trial. It stays in the same place. So kind of the take-home message from this is similar to what you said, Jonathan. There is a high-dimensional dynamics, and it's funneled into these two attractor states. And this is different from what they used to model this type of dynamics, where they kind of thought of this classical two-dimensional, two populations, where you have only one dimension, which is only the choice. So the only way to flip it is to go in the direction of the choice. This is not what happens. The separatrix, in some sense, is in high dimension. It's a bit more complex than that. And this is the data. This is already in bioRCAP. This is the data by Kayvon. Kayvon is doing this from Carr's group. He's now also a scientist at the Allen. And Kayvon has developed these techniques that also our Finkelstein is using of perturbing single neurons. So then you can choose the single neurons according to their choice. And what he found is that, as the model predicts also, the selectivity along the choice dimensions, right? This is this axis of the perturbation that you do using optogenetic perturbations, have almost no effect on the behavior. Your ability to flip the decision is mainly zero. And this is not the case for residual dimensions. How we define residual dimensions is a different story, but he has a different way to do that. But you see that if you perturb it along different dimensions, then you can flip the decision. So there is an effect of the behavior. If I'm going back to your question, so there are several examples. One is Arseni. Arseni did in his postdoc a specific perturbation. So he activates somatosensory cortex, and then it was a go, no-go test. So it activated the somatosensory and then recorded in the ALM. And then the perturbation was another activation in the somatosensory. So in a way, this is an activation in the direction of the stimulus. And this can flip the decision at the beginning, and again, not later, like we show here. The other perturbation that people have before is just a channel rhodopsin for the inhibitory neurons. You activate all the inhibitory neurons, and you just decrease the activity of the neurons, and you just you're doing just a chance. So you're not really flipping the decision in these experiments, if I remember correctly. Okay, so the take-home message from this part, we have these residual dimensions. The way we define them, they are orthogonal to these coding dimensions, and we find them through the training of the network, and it affects the neural dynamic and the choice. Neural activity during decision making span really large number of dimensions. It's not a two-dimensional model like the typical model that we have in the field. Neural activity relaxates onto one of the two attractors at the end. Or in other words, this model that we have is really not what we see in the data, and the data is a bit more rich than that from the sense of view of the dynamics. Okay, so yeah, good. We have enough.
Could you give some intuition of what?
So
there are two attractors or more.
So what we think happened again is that so we're still analyzing some of these networks to really understand the mechanism. What we think is that there is kind of a combination of the two models that people think are related to this data. One is this kind of these attractor states. So there are these attractor states, we think. And the other one is that during the decision you have kind of a sequence. It's not really a sequence. A sequence is something that you model in these models kind of with the shift metrics, like this dimension is driving this dimension, driving these dimensions. We have it from the connectivity metrics. It's not that. It's more complex than that, but we think that it's something along this direction. That you have modes in the stimulus, they are driving other modes until you get the choice, and then the choice is kind of separating the trajectories to the right or the left attractor. So that's and again, at the end you'll get to that, but the separatrix in 10 dimensions, and we are interested in calculating that, because in 10 dimensions you can calculate the separatrix with all kinds of methods. The separatrix is a bit more difficult, a bit more complex, because it's not a two-dimension thing that you just build. So the way to go from here to here is not like that. Maybe it's like that. Right? It's not the easiest way to go.
So this idea would work on this kind of delayed memory task, not on evidence accumulation, which is more classical decision task.
What do you mean won't work?
I mean, if you need to accumulate evidence along and integrate evidence before to a decision, so you can't make just a general trajectory. You need your evidence to push in the right direction at every point of choice. So this is would be a very different model, like the Montesquieu case, which is a very different model from this delayed response when you just need to memorize or make the decision and then
the dynamics is not the same, I agree with you. I'm not saying that it will be the same underlying mechanism. The concept the task is different. So of course the mechanism, why do you expect it to be the same? You have accumulation of evidence that you don't have here. And Tanya Engel has a paper on that where she's trying to do something in this direction on the Mendes-Cecilo stuff. She's arguing it's not line attracted. We can apply it to that, and I think we'll do much better, because what she explained with their model 10% of the variance. And I think I already convinced you that even if you expect 90% of the variance, you might throw away dimensions that are really important. And so that's what we think is going on there. I don't know what is the mechanism. Yeah.
I mean, it's a good question. So the choice dimension is one dimension if you have these two attractors, which results in a very high dimensional null space, right? So where are you doing the perturbation exactly? In the residual perturbation, like in a 10-dimensional space, if you have one-dimensional choice space, are you like perturbing in that 9-dimensional null space, or are you specifically choosing some dimensions? Like,
so let's break it to two. the choice dimension in 1 dimensions. We have n-1 dimensions, right, in general. Here we are fitting a p-dimensional system. So p is much smaller than n, so it's 10. So we have, if we have 1, we have 9 other dimensions. Still have 9. What we did here is just, what I think we can do is to calculate the separatrix in these 10 dimensions. We can calculate the dynamic. I think so. Like numerically, there are ways to do that. And we didn't do that. It's not that easy. But at least we can perturb it randomly each one of the 9 other directions. That's what we do. In a systematic way, but in each one of the dimensions. You can choose some combination of them. We didn't do that. And so in a way, these dimensions, they are, we find them through the fitting process to the data. These are these other dimensions.
But there still has to be some sort of ranking, right? Like in some space you would get more of an effect, while others you wouldn't, and so on.
I agree. I don't know the answer for that. We still we don't know the answer. Yeah.
So is it really n-dimensional or 10-dimensional, or is it like decomposed into, say, different population of neurons that some are actually doing the choice in 2 dimensions, and the other, what you call the null, are actually driving this dynamics?
So we just started to touch it, and we have a way to extract populations in a way. And there are ways in this kind of type of networks to extract different populations, kind of clustering in a way this how this mixed selectivity that you have for all dimensions. Like there is a systematic way to do that, and we can do that. We play with it a bit. I would say that if I remember correctly, and again, the problem here is that you need to be convinced that if you train multiple networks, you get solutions that are robust or invariant or something like that. But we started to do that, and it seems that really there is the population that is really important for the decision is not that large. But I don't know the answer for that. But it's true. It's a good direction that we are exploring in a systematic way. Yes.
So if we're in the so can you give an answer for the Charles, the robustness result? Because you said that it was just a random choice of the silencing. If this was activation, you could get, you would find that it's not robust, or you can answer your answer.
I don't know. Okay, so I'm not sure what is the question. So again, I will answer in a.
You have a perturbation, but once it's over, you get right to where you were.
Yeah, so the trajectories are stable. The trajectories are stable.
Showing that you can fit them.
The other direction, so the perturbation that Shaul did, and not Shaul, but the perturbation that people did in the Svoboda group are just homogeneous perturbation. You are perturbing all the modes.
So it's kind of a random direction.
It's not a random direction, it's a homogeneous direction. It's a very specific dimension.
More attractive.
All right, let's continue later. I really want to talk about that. I can skip this, but I really want to talk about that because I think we have a cool method here, and I want to hear your inputs as well. So decisions are at the level of single trials, right? When I make a decision, it's a single trial thing, and we want to understand if these residual dimensions, the fluctuations in these dimensions, are what actually generating error trials, or what generating decisions at the level of, like error trials and decisions at the level of single trial. So what we did, we extended our model to single trials. How do we do that? We take exactly the model that we had before, but now we're going to drive the network through this coding and residual dimensions. So it's just the inputs, and it's only a bias term. And so we add another term that we fit, which is a matrix which goes the number of trials and the number of latents that we have. So we are training to single trial now, exactly what we did before, this low-rank connectivity matrix, plus a bias which is depending on each latent, and it changes over trials. That's what we do. And then we need to define a single trial loss. And what we do, I think, I really like it. I don't know if that's, I'll be happy to see what you think, but what we do is something like that. So, you can look at neurons over time as we did before, and now you have also trials. So this is kind of this tensor of activity. You can average over time, over trials, right? That's what we have over here, and that's the trial average that we did before. That's the loss that we had before. Now, you can do other stuff, right? You can, for example, you can average over neuron, So then you get kind of the average activity of all the neurons changes in time. We have all kind of dimensions coding, which we think is important, so we can average over them as well. And they will change over time and over trials. And so we can do averaging also over projection. It's kind of when we do this mean field analysis, we average over many neurons, and so it's kind of this. And we can also project average over time, because we can do all this average. So we have neuron over trials. So we do this type of averaging of this activity tensor, and then the total loss is just the trial average losses before. Now we add all kind of projection average loss, as we said before, and also this time average loss, which is kind of what happens over trials for each neuron. And what we train is only the biases. That's it. Just an average bias for each neuron that changes over trial. And note that we never train a single trial. There is no loss on the single trial on the neuron at each trial. There is only all kind of slices of this stuff. But there is enough information here to reconstruct the single trial. So we are not fitting a single trial, but with this averaging, and we kind of, at the end, I'll show you examples, we kind of can predict single trials of single neurons. So here's how it works. Here you see single trials. This is projection of a single trial, the projection over the stimulus direction for left and right, projection over the choice, the response. Again, the wider line is the model. You see over trials, by the way, to see how non-stationary the neural activity is. Neurons are drifting, right? The bias is drifting, the average activity is drifting. Again, 2 colors here is for the model and the data. Maybe it's not easy to see the gray one, but it's there. And finally, look at the single trials. I never fitted single neurons at the single trial level, but still we can capture this, this nice bump that we see over here, probably because there are correlations with other neurons, and kind of with this slice we take it, we get it. So then we can skip these slides and go to the other slide because I think it's interesting. And what else we can do? We can kind of ask ourselves, all right, what is generating an error? So we can take a fit activity to error trials and to miss trials and to correct trials. And you can think that maybe in this latent space of these kind of biases, this trial over latents, maybe in this input space, there is a direction that best separates the hit trials with the miss trials. So maybe there is an input that I can give to the input, to the network, just an input, a bias to in this latent dynamics that can flip the choice. And that's what we looked. We looked at this matrix that we fitted to the biases, and we looked for the direction that best separate error trials and correct trials. And you see it works quite well. This is the left hit and miss trials, and this is the right hit and miss trials. So this is really a direction in the input cells that is informative of the error trials. And now we kind of have a generative model for error trials, right? We can generate now. We know the direction that best separate. This is this axis. Again, don't be confused. It's not the directions in the neural space as the choice, etc. It's a direction in the input space. P equal 10, so 10 dimensions. And you see we can perturb it and we can flip the choice of the model, right? It's not the choice of the model just by choosing the right direction in the input space. And you can ask yourself, all right, who are these dimensions? Are they the input to the coding dimensions, for the residual dimensions? So it turns out that you really, without any inputs to the coding dimensions, you can still, the projection of this error choice, either for the left classifier or the right one, and you have a lot of, the projection is large for many of these residual dimensions. So in many of the residual dimensions, I can actually span this error axis with the the input to the residual dimensions. All right, I'll skip about the multi-regional, but practically the story is similar. We do a multi-regional, and the communication between the two areas, this thalamus and motor cord and ALM, is through residual dimensions. You do perturbation in thalamus, you flip the decision in the choice in ALM, and only if you do this perturbation in the residual dimensions, the choice dimension is more stiff. So the take-home message, we built a data-driven framework to dissect this coding and residual dimensions in the data. We fitted it to neural activity, and we found these high-dimensional trajectories converging onto two selective attractor states. And single trial modeling allows us to show that these errors, are probably driven by fluctuations along the residual dimensions. And this residual dimensions, so that's the two two areas, and these are these residual dimensions that are flipping that are communicating the way the ALM and thalamus are communicating, or at least with this choice direction, but also with this residual dimensions. And yeah, with this again, thanks to the group, collaborators, and to the funding.
You know, the questions are it's only 1, 2 questions.
So what do you think is there in this residual subspace? Like the space of residuals, I mean, clearly it's not stimulus, but it does affect the stimulus. So what do you think is happening?
Yeah, and there are options to check that we are checking. One is kind of people like a ramping mode, kind of the urgency to react is kind of neurons are ramping their activity up and down. This is kind of a dimension that might be related. We're not sure, but maybe there is kind of, you can add another, I don't think that that's the case, but you can kind of add, let's see, we define it based on time, right? So we define the sample here, the choice here, the response here. Maybe there is something over here. So there is another dimension that you can capture, and maybe this is early delay direction that is driving. Maybe what we think is that there is kind of a sequence that, or in a way, it's not a pure sequence because we've seen it from the training. It's not that you have one dimension goes to another, get to the choice. It's not the case. That's kind of a simple model that they have in Kavon's data that we think it's not what happens, but it's something along this line. There is some kind of non-normal kind of transient activity, and these two trajectories separate, and I don't know exactly what is how the separatrix is built, but it's kind of, these two trajectories separate in this high-dimensional space, and how do you move from one trajectory to the other? There might be rules there, I'm not sure. Like maybe there is some consistency, but we still didn't explore that.
I'm not sure I understand how the things develop with the residual because it puzzles. It's a puzzling idea. So maybe to clarify, we can go back to a different kind of experiment, like Newton experiment, where you perturb the system, and your perturbation changes the decision making of this element. That the drift of the dots are not here but there. Will your model explain that data for the perturbation of the residuals?
So I don't know the results.
Because there we always thought it's because the neurons are clustered and have the same directional motion direction selectivity, which is
stimulation was aligned in the coding direction. I mean,
when it was aligned.
No, the way the Newton's work managed to perturb is that, as far as I remember, the stimulation was of cells. The electrode was stimulating near cells with a specific preferred direction, and that led to a bias in that direction, which is different, I think.
It's different. Of course, it's also if you really perturb in one direction, if there is some barrier, you will pass it.
But this was his concept. The neurons sit there. There are thousands of neurons sitting there. Some of them are tuned this direction, but many have many different. What Ran showed us, like this classical drawings of tuning curves, is not what all the cells do.
But this is not in the motor cortex. In MT, the cells are aligned in the same direction.
That's what I asked about.
Yeah.
What do you mean in MT the cells are
because
aligned in the same direction?
they think
The column?
Yeah.
Then there are columns for the directional selectivity.
I see. And then you perturb this column and you flip the decision.
Yeah.
That's what you're saying. Or
So
that's what you did.
So what I understand from your half an answer to Elon, before we interrupted you, is that a strong enough perturbation in the choice dimension will still be able to flip the choice.
Yes, I think so. Yeah, and maybe not always, but at the end, it's kind of how do you there is kind of two decisions or attractors, and there is some separatrix in this, I don't know, 10 dimensions. How do you go from one to the other? Of course, if I perturb that strong, I will be here, right? So the dynamics is not important anymore. If I just move you from here to here very strongly, you'll be here.
But in a way, I think, I just want to add to Elon's question. I find it, the terminology of residual, it's not a residual, it's this unknown dimension, the thing that we don't know what it codes for, and so it's not like just in a way it's a kind
You're
of...
right. First of all, if I just do a random perturbation in this n dimensions, it won't work. Yeah. In a way, the coding dimensions, because I make the network find these coding dimensions, I kind of find also the residuals, right? It's not just a random vector in space. If I just randomly take n dimensions, just one of these n dimensions randomly perturb, it won't work. It's part of the dynamics, right?
Yes.
We needed to explain the neural variance, the neural activity. So it's parts of the dynamics. Why this is more important than the choice, I don't know. Maybe it's not robust. Maybe we still need to do some work. But it's part of the dynamics. And I used to call it non-coding. And I thought about it then. Well, it is coding. There is kind of information on the task, and they are residuals. This is why I thought it's residuals. It's the residuals, and they can of course code other stuff. They can code the movement. They can code whatever. But they can code time. I don't know what they code. That's this question that Lilach has. I don't know.
It's clearly that they code something.
Of course. They are part of the dynamics and they are important. I don't know if they are coding something.
The naming residual is a bit, I know it's like super fashionable to talk about residuals, but...
I don't know. I thought, again, I think residuals are the rest, right?
But it depends. You define what is sufficient, right? So then
you're right, but also when you do the dimensionality reduction,
right,
it's on
exactly.
you that you are defining your coding dimensions, right? I'm just saying that the one that you throw away, that you think explain 5% of the variance and not important, they might be relevant because they are part of these dynamics, and everything is dynamics, right?
Yeah.
Yeah.
שכל הקודינג הזה, מה שהראית, כאילו יכול להיות שיש דינמיקה של מה שקורה. ברוב הווריאנט זה נוירונים שיודעים לקרוא את הדאטה ולהשמיע לנו. מדהים שיודעים לקרוא את הדאטה ולהשמיע לנו. רק עושים רידאוט. בסרט פרטוביישן זה לא משנה, הם קוראים דינמיקה אחרת שאתה לא השפעת עליה. אז כל מה שאתה עושה בפרטוביישן שלך רק משפיע על משהו שקש לחזור לדינמיקה שהם קוראים מהרשת.
זה גם לא חלק אתה אומר שלא חלק
מהדינמיקה? כן.
אז מי אתה רוצה לקרוא?
כי הוא גם חשוב, הוא מסביר 100.
לא, אני אקח דינמיקה שבה יש לי רידאוט נוירון, ועכשיו אני אעשה להם סיבוב רב נמדי, ואתה מוצא אותם. אבל בסוף המנגנון, אז הם סוג של רידאוט.
לא הבנתי מי הם...
ורגע, ואינטרבציה של...
אתה עושה אינטרבציה לקטע.
כלום לא קורה.
נכון, בסדר. זה מה שהצ'ויס הזה, וזה רידאוט של המערכת, זה בסדר. אבל הוא לא חלק מהדינמיקה, הוא לא מייצר את הדינמיקה שמובילה לצ'ויס. זה כל מה שאנחנו... כן, הוא רידאוט.
אתה הולך עכשיו,
גם כשאנחנו מסתכלים, עושים להנליז על המודל, אחרי שהוא למד, אז אנחנו... שוב, אנחנו רואים שיש הרבה מודלים אחרים שדוחפים באיזשהו מובן, והוא קצת הרידאוט. המודל הבסיסי יגיד שזה רידאוט. יש כזה סיקוונס כזה, אתה מכיר את הדבר?
אנחנו נעשה החלפה של הסדר, אנחנו נדבר זמן קצר ואני אביא אותך לרדת, בסדר? אני לוקח אותך איתי. כמה שקטן, כי היה 2000, אמרה לי שאתה נמצא לך, לא תראה, דינמיקה עם
זוכר.
מה דימנשיונליטי
שמה? של
הטאסק, של הדינמיקה. אז גם יש לנו את זה לדינמיקה כללית, וגם מתחיל לראות לשחק כל מיני משחקים כאלה של ממורי ופליפ וכל מיני משימות כאלה של
העלית את זה רק בכמה סליידים אז שאתה מתכוון.
לא, אז זהו.
אני לא יודע, כאילו, נתת איזה משפט. בכנס ראו את ה-P, שזה היה נראה לי מעניין, אבל באמת לא נתת על זה, לא היה על זה כמעט כלום, אז אני לא זוכר, הלכתי משהו. בוא נדבר, כן.
לא, זה מעניין,
אני אפילו לא יודע איך.
אנחנו יכולים לעבוד בכמה מקרים באמת שהרקע האופטימלי הוא גבוה יותר.
הרקע האופטימלי
הוא גבוה יותר מהטסק דימנשיונליטי. יש לנו כמה הוכחות